The keynote presentation of the day that is going to be delivered by Professor Matthias Mann. He comes from the Max-Planck Institute of Biochemistry in Germany, where he has the proteomic and signal transduction group. And also he has the clinical proteomics group in the Institute for Protein Research in Copenhagen. So I think that if you are a mass spectrometrist or close to mass spectrometry, you already know who Matthias Mann is. There is a long list of honors and prizes that he has received. So it suffices to say that he has 310,000 citations and an H index of 260. So I will stop here and let him tell you about the latest advances in proteomics and mass spectrometry. Thank you for being here.
Yes, I'm very happy to be here. Indeed, I will tell you about the latest and greatest. I'll focus more on the applications rather than the technology. I will also try not to make it too long because of the advanced hour and also because I got up 40 hours ago in Chengdu, China, and I haven't seen a bed since. So this is our institute in Munich, and we do proteomics, so it's a contrast to just the last lecture. So we focus on proteins, but of course all the biomolecules work together and cannot live without each other. Nevertheless, there's some interesting facts about a cell that I want to come back to, and just sort of tease it here. And that is the fact that we have just 2 copies of each gene from your mother and father, and just also very few transcripts, especially in post-mitotic tissue. And then we have many more proteins for every transcript, or even if there's no transcript and the cell needs that transcript, then it still has the protein, and that may persist for years. And also you have full, all the proteoforms and the spatial organization of the proteome. You have that in every cell for a long time. What we always say in proteomics, that proteomics are the functional actuators, and their direct measurement is the closest you get to function. And then, of course, we are always behind with our technology, so that's incumbent on us to make the technology as powerful as possible so that one can take advantage of these theoretical advantages that the proteomics level has. So then this is what you can do with proteomics. I mean, you can do many more things, but these are some of the main categories of bottom-up proteomics. So the one is global proteomics, where you try to measure as complete a picture of the proteome of a given system as you can. So in a single cell type, maybe 10,000 proteins would be a good, almost complete picture. And so this is the equivalent to transcriptome measurements. And then you can do protein interaction networks by pulling on a protein or any bait and seeing what other proteins come down with it. You can also do that with drugs or pieces of RNA or DNA. And then we and others have looked a lot at post-translation modifications, and one can now measure tens of thousands even in like 20 minutes. And then the current hype is single-cell proteomics. So I'll have a little bit of story about that and how that's same or similar or even competitive with transcriptomics. And biggest theme will be clinical proteomics of my talk today. And we've been focusing on that area for the last 5 to 7 years, more and more. So I want to start out by a recent story that got published this year. And this is also to show you what proteomics can do, and in this case uniquely could do. So the story is from the Scandinavian brown bear project. So these are and with the hard people from Munich here, and this is the postdoc from my group here. And the question that the Brown Bear project wants to address is why the bear can sleep for half a year and not have any of these problems here, even apart from these here. It also gets, in the winter, gets plugs in the brain, and then they miraculously clear up again in the spring. So how does it do these things? So that's an ongoing thing, and if this was playing, then you could see a very slow and irregular heartbeat. So yeah, focusing on these heart problems, Johannes then looked at the plasma of the brown bear, but there were no real big differences in the plasma of brown bear between hibernation and non-hibernation. But when we looked at the platelet proteome, there was a smoking gun, and this is this HSP47, so that's also called serpin, and it's the surface protein on the platelets, and it kind of makes them sticky. So that sounded interesting. And then there's a pig model here where of rest, and also 2 human models here where the same trend held as well. And then in the paper, there's a proposed mechanism how this would happen. And so this seems likely to be at least one of the mechanisms that the brown bear has to avoid these aggregation problems in the blood. That could even be, it's not so easy to target this, but there's several activities now how to perhaps target that. So, and I also like this example because the platelets don't have a transcriptome to speak of, so you couldn't have seen this by transcriptomics. So, yeah, and this also made the news, even almost the German, made the daily news in Germany and elsewhere. Okay, so that was just kind of a warm-up, and this is what our group tries to do. So over the years or decades, we've always trying to push the technologies. So this is a technology where most of the results come from in my talk, and this is so-called timsTOF. We already heard about it earlier with the glycoproteins from Sonja Hess. So this is mobility here, and quadrupole, and a time-of-flight analyzer. And this is a particular principle that we added here, where we stack the ions and pick them off one after another, which gets you like an order of magnitude more speed or sensitivity. But this is just an example of technological advances. And then here, this symbolizes the clinical part, and in particular the symbolizes going directly from biopsies in FFPE format, which is the thing we're focusing most of in the latter part of the talk on. So just to be on the same page, we use a kind of a classical bottom-up workflow where we always, so we have any source of protein, so this could be anything from plasma, for instance, to the microbiome or anything like that, and then you have to solubilize this and then digest the proteins by trypsin into peptides, and this is now, because we can analyze them better, and this is now done in an automated fashion for instance, we use this Agilent Bravo, but it can also be an OpenTrons or something like that. So this is pretty well under control nowadays. And then we have the liquid chromatography, so that used to be the most finicky part and maybe still is. So we have standardized on this EvoSep instrument, which I will also talk about in the single cell context more. Then as the columns, we use either these PepSeps that sort of came with the instrument, or now for the ultra-high sensitivity, we use the ion optics, and then there's a column that's interesting for the future, I think. So this used to be its own Belgian company, but now it's part of Thermo, where you actually print the, you don't have beads, but you print the columns with a C18 material, so they cannot move, and as a result, this is very stable, and it would be the same in any lab. So if you want to go to the clinic, this has very good properties for the future. And then this is Evosep that makes a preformed gradient that I'll talk about later. And so we've in the last few years worked a lot with Bruker and still do with this timsTOF that I just showed you. We also have Cyc-Xenotoph. So they haven't pushed so much into proteomics yet, but this has an interesting EAD fragmentation method where you can not only fragment them by collision in the gas phase, but with transferring an electron at a variable range of energy. So this is interesting for small molecules in glycopeptides and so on. And then the latest buzz is the Orbitrap Astra, which has been just introduced by Thermo at the ASMS conference a couple of months ago. And we have a number of them already, and I have just very few results to show on them, but I'll be happy to expand in the discussion, why that instrument could be interesting. And then you generate a lot of data, and in the past, and people still use that a lot, is Max Quant, but Jürgen Cox, who wrote that in my group, he has his own group now, and we have taken a new page here, and we have this alpha universe here, so alpha PEP search engine, alpha DIA for label-free quantification, or for data-independent identification here, that's coming out hopefully by the end of the year. And right now we nearly always do data-independent acquisition these days, and we use mostly DIAN, but also Spectronaut for phosphopeptides. And this is an effort from our lab, but this symbolizes making a sense of the data at a higher level with a clinical knowledge graph or things like that. So then I have a short story here about interactomics. So this is an area where Mass-Spec has been quite successful. And so here there's two stories. One is the yeast interactome, and so more about that. And here, this is together with the Chan Zuckerberg in the BioHub in San Francisco. They did a lot of microscopy, and we contributed the interactomics, and that came out recent or a year ago. So the way they did this is tag by CRISPR, tag the human proteins with a part of the GFP, and then the cell already expresses the rest of the GFP, so then they snap together, and you can do microscopy, or you can do pull downs, and then you have both the localization and the binding partners of a number of proteins. So this by no means complete, but we think it's quite high quality, and then you can also start to do machine learning embedding for where this is in the cell and also what it interacts with, especially as this gets more complete. So and you can do this now fairly quickly and with high quality. And here, this is the yeast interactome. So there were classical efforts here 20 years ago, and I was even part of one of them. And this also illustrates how the technology has progressed, because that time it was a team of 30 people for more than half a year, but we only produced interactions for 1,000 baits out of the 4,000 yeast proteins. And we used, for every pull down, we needed like 4 liters. And so that was this whole team. And now, with all the improvements in technology, we thought we could do this both faster and much better. So we only need 1.5 milliliter per pull down, and that's even too much. And this is done by a single PhD student now, this whole work. So then a PhD student, Andrei Michaelis, he did this in 96-well format and like this with the Ivosep and the Bruca, and this just runs every day for a while with 60 interactomes per day and with a very high quality, as I'll show you. So you can see this here. So these are epigenetic complexes where, in principle, when you have a stable complex, you should be able to pull down on every member of the complex and get the whole complex, right? And that, so if you have the members of the complex here, that should give you rectangular profiles for all the members of the complex, like these orange lines, and it should give you nothing here if they're not in another complex. And that's kind of exactly what happens here. Same with this one, and same with this one. So that so it's very high quality, and even some complexes are untaggable, because when you put the tag on complex, they don't want to go into the complex. But still, we get those complexes by a correlation with the other baits. So those we have as well, and also membrane complexes and so on. So we believe this interactome is actually complete for a static interactome at least. So we find back all that's known, and it's not like we find like half the complex in these were unknown. So this is more filling in the gaps in some ways. But still, we find basically textbook knowledge back in one experiment, and these previous efforts were criticized that they were noisy and they don't overlap with each other. But people made a consensus interactome, and we cover that consensus interactome and add a lot more interactions as well. So now, and this is also much better than the binary interactomes that keep getting published. So then we can look at what are the network properties of the yeast. So we can say that almost all the proteins in the yeast are social. So we had a very small percentage that we didn't find interactome partners, and then the reviewers made us do reverse tagging on them, and that even halved again. So there are very few that we don't see interactors for in the ground state. And then there's all kinds of network interactions, like what is the mean distance between two partners, like the famous 6 degrees of separation in human society. But then Facebook said, no, it's not 6 degrees of separation, it's 4.5. And then we also actually find in the yeast interactome, it's also 4.5. So this is maybe some underlying network property that's the same for Facebook users and the yeast proteins for some reason. And then what you can, of course, do is if you have even a known complex, but now you have a new member, you can put this in alpha-fold multimer and then see what it does. So for instance, in this CK2 example, there's a new, we found a new protein, then put it into alpha-fold multimer, and it came out not with one structure, but with two structures for this extra protein. And then we followed it up, and actually both structures exist, so they are regulatory versions of each other. So this is not like, this doesn't happen for every complex, so only some of the complexes works, but I think that's kind of the way, and this will be an elegant way, right? Because pulling down on the bait, doing the mass spec, that's literally minutes now, and then you can put it into alpha-fold multimer or similar and see what it comes up with now. So it reminds me of the extinction watch before that we saw. But to some degree, we can then guess what the structure will be. Okay, so now I'm switching to clinical proteomics. And we basically have two things here. So one is the body fluids, like blood, most famously, but also urine or CSF. And especially the blood, the big promise will be that it could be a universal phenotype for the human, and also most of the tests in the clinic are either small molecules, but or plurality at least is levels of proteins in the blood, like C-reactive protein, for instance, CA125. And the promise of Mass-Spec would be that we can get this all together in one go and find many new interactions or many new biomarkers. So that was a promise, but it was hard to deliver on the promise for technological reason and also for many other reasons. And that isn't only the case for Mass-Spec, but also if you remember Tyrannos and Miss Holmes, she's actually in prison as we speak. So people were hyping this even to a degree that they had to go to prison. But then we wrote this, so we kept out of all this before, but we wrote this review a number of years ago. And since then, I'm happy to report that we actually do find markers. So I won't talk about it today for time reasons, but we're particularly proud of this one here, where we, and it's also a big organ, so it stands to reason, but so we can find by Mass-Spec now better markers for early stage fibrosis, which is the stage where liver disease becomes irreversible, than any of the existing methods like ultrasound-based or enzymes in the blood-based. So that's coming along, and we and others are busy with that. So I'm happy to expand on that in the discussion. And also, we just written a review, if you're interested, in molecular cellular proteomics, where we call this the end of the beginning. So it's now for real, and I do think it will be very useful. I mean, there's competition with technologies like Olink and so on. I can expand on if people are interested in. So now, the rest of the talk will be about single cells and single cell types in the tissue. So that's, as I said, that's the current area of excitement. And there was a Nature Message special issue earlier this year. And we also wrote a perspective where we said, okay, if this should be biologically relevant, then it would be good if we could identify like 4,000 to 6,000 proteins in a single cell. And this is here the trajectory of how many proteins can actually be identified in a single cell. I think I'm even on record to say not so long ago that single cell proteomics will not happen in my scientific lifetime, but I have to admit I was wrong. So this has shot up dramatically and is still shooting up. So much faster than the bulk proteome was before. Because inherently it relates to my first slide that there are actually quite a number of proteins even in a single cell. But also we have to remind ourselves that single cells have quite different sizes. So on the one extreme, a resting T cell, for instance, has 20 picogram of protein before it becomes activated, and then a human oocyte or even a chicken oocyte, they're quite gigantic in comparison. So what people in proteomics like to take is a HeLa cell, so there's 250 picogram of protein material in it. So our entry in the single cell proteomics field was, this was also a bit like the plasma controversial, the first work, to say the least. But we worked with Bruker, so this is a sort of a cross-section or taken off the cover. And what they did together with us is like put more ions in. So that gave us a factor 4.5 in sensitivity. And then we worked on this chromatography, and I should say I have a conflict of interest because I'm an investor in this Danish company here. So the principle of this EvoSep is that you preform a gradient with these low pressure pumps. So you pump through a tip here where your peptides are, and you pump through with this gradient here, and then a makeup gradient, and that together forms the gradient here in this capillary, which then gets pushed out by one pump. So this is the only high pressure pump, pumps it over the analytical column here. So this was made like that for clinical stability. and runs normally at 1 microliter to make it applicable for the clinic. And then we have two advantages here. So this was never meant for single cells, but we have this tip here, which now can serve as a reaction vessel for with a very small volume for the single cells. So we can elute them here in 20 nanoliters, and they are safely stored on this C18 and Teflon material without being absorbed. So that turned out to be a key advantage also for other things. And then the other thing is here. So this is the whole setup in 394 well placed to have a small volume. And then we can do 40 single cells per day here. And this is pushing out the liquid at a small flow rate. So normally it's 1 microliter, that's the blue trace. But when you push it out with only 100 nanoliter, then according to electrospray theory, it should become 10 times more sensitive, and that's exactly what it does. So then we have the 5-fold and the 10-fold. So now we have 50 times more sensitivity, and that indeed allows us to look at single cells. And and so here is the quantitative comparability. So this is bulk proteome against bulk proteome, so the rank order of the abundance of the proteins. Then we go to only 8 cells, so it pretty much holds up, and even for a single cell, this holds up. So the quantifying one single cell against another still gives you a very good rank order. And then we can also do a drug experiment on these single cells. So this is a classical blocking the cell cycle and releasing it experiment, which we did on the cells. So then we measure the single cells, and this is done afterwards. So you can see in these stages we get more proteins because the cells are larger, and here in interface we get less proteins. But clearly we can see from the proteome, we can see these cell cycle stages. And when we quantitatively compare them against each other, we also see cell cycle markers that are different as they should be. So, and if you can do this for cell cycle, you could also do this for drug treatment or other drug treatments. So then we can compare this to transcriptome data of the same, of a similar situation. So these are two different single cell transcriptome measurements technologies. So they correlate amongst each other, amongst each other here and also to each other, and not so much to proteomics. That was expected, but this I certainly didn't expect. So in the proteomics, which is a sort of new to the game, the CVs are much smaller, like 17% or 30% here. And also the latest transcriptomics technologies, they have a very large variation of the transcripts. And why is this? That relates to this first picture I showed you in the introduction. This is because the cell, this is even for cycling cells, right? But even the cycling cell has usually just maybe 1 or 2 transcripts for a gene. So then you have these counting statistics. If you look in single cell data, you have for a given gene, you have 0 transcripts or 1 or 2 or 3, let's say. And then if you average that over 10,000 cells, you get some quantification. But this is not the case for proteomics because, again, we have typically hundreds of copies and we don't have this shot noise. So I think this is a long-term advantage of the single-cell proteomics. Also, maybe later it becomes possible to measure modifications even in single cells or isoforms as technology advances over the next years. And this you would get in single-cell proteomics, but not transcriptomics. Okay, so but what we're more excited about is the spatial aspect and combining the single cell type with the spatial aspect. So this is the usual motivation for that. So for instance, when you have tumor, we would ideally like to start with FFPE material, so they are very amenable to proteomics. More difficult with transcripts here. And then we would like to not like homogenize the whole tumor, but we would like to know what is the proteome of these individual cell types, and for instance also the immune cells. So then this, this is then putting some things together. We call this deep visual proteomics, and that stands for deep learning. No, okay, this is first what people did before. So before, people could also analyze already for 10 years FFPE samples, but then they would do, including us, scrape everything off, or some macrodissection, or in this case microdissection, and then get some proteomics. But now we have mixed up all the cell types, and that will make it much more difficult to get some biological meaning out of it. Yeah, plus it was manual, which can be tedious. So we've been active in this field for a while, and done this bulk or compartment resolved, also with good results, but still that's not what we want. And what we want instead is this cell type resolved tissue proteomics. So here it comes. So again, we start with the FFPE, of which there are maybe 1 billion of them in the world. And they don't age. So we've analyzed decades old of these paraffin blocks. So now, so the deep stands for deep learning, and visual stands for imaging. So that's this part here. So now we have our FFPE material, and we can use any kind of imaging here. So anything from simple H&E to multiplex imaging, and what we need is a marker for the cell surface, for the plasma membrane, and to know where the cell boundaries are, because we're going to cut them out by laser microdissection. And then, so that's the one machine learning part, and then the other part is classifying these cells into one homogeneous type in the same area. And I should say this was spearheaded by Matthias Mann and Fabian Korscher also, and he has his own group in Berlin. He's got a PhD grant and AI partner in Hungary as well. So now we have, so what we do then is not cut out a single shape, but we cut out, let's say, a couple hundred shapes, but they're of the same type, so we preserve the advantage of having the cells the same cell type and shape. So these will be collected together in the same well here, and that also gives us the throughput that we need. So then collect them and analyze them like I showed before, bioinformatic analysis, and then we hope that this can lead to cancer-relevant discoveries, or even be a decision support for a given patient here. So that was published a year ago, and here's an example. So we have here, so this comes from our work on ovarian cancer in collaboration with Ernst Lengiel, and this is from Lisa here. So what we have is, or what we want to characterize as an example, is the epithelial layer of the fallopian tube. So the fallopian tube moves the egg, and for that purpose it has two cell types. It has the ciliated cells that move the egg along, and it has these secretory cells that produce the mucus. And these here express FOXJ1, this transcription factor, and the other ones don't. So we can stain for that, and we can teach the AI in a couple hours what is the one cell type and what's the other, and then it can cut them out, like shown here. And then we do proteomics on the one cell type and the other, and this separates here in PCA. And this is the protein atlas from Stockholm. So we find this transcription factor never in these cells and always in these cells. And these are other examples, markers for these two cell types, which that's also the case. And then if we do bioinformatic analysis of these versus these, they tell us exactly the functions that I just told you about, like this one cell type is ciliated and the other one does stuff that secretory cells do. And this is also called biological enrichment. So the function sort of jumps out at you because you have pure types. And this shows, and this is from the same paper, that we don't need to stop at single cells. We can also cut out just nuclei, or we can cut out whole cells and compare them to the nuclei. So for instance here, this is just from cell culture, but the AI can see 6 different classes, and we can ask, what's this banana-shaped nuclei class even doing? And here we can see what we cut out. And then if we ask this question that I just asked, then the bioinformatics analysis tells us that this has something to do with EMT, so biological functions that are associated with EMT in cancer, they come up. So maybe this, but this is just cell culture, but this shows nevertheless the principle. Okay, now this is sort of a real cancer example, and this also shows what was wrong with previous ways to do this. So this is the typical situation where you have the tumor here and you have the normal tumor here, the normal tissue here, and the normal tissue has this finely resolved cellular structure with many different cell types and it's doing what it's supposed to do. And the tumor, in the tumor, one cell type predominates. And now you have 2 choices naively. So you can compare this with this, but now you're just measuring the difference between different cell type compositions and not what's wrong with the cancer cells. Or you can homogenize the tumor and compare that to 200 different patients. But now you have the difference between the 200 patients. And the beauty here is now, so we can now classify these cells, and we can look at the cells that are the tumor cells here and compare them to the same normal cells or precursor cells, if you will, here, right? And then now we just cut out these and compare them with here. So now we have for one patient what exactly went wrong, or how are exactly the proteomes and thereby the functions different of this tumor. So this is shown here, and these are just replicates to show that it's quantitatively reproducible. But now we can exactly compare the proteomes of the cancer cells to those same cells over here, and then we can see what was upregulated. And here there's many proteins upregulated, but for instance there are these immune relevant proteins, but also this Sark kinase, and because it's a rare cancer, this wasn't known. So naively speaking, if this hadn't been an archival sample, you can now treat this patient with a Sark kinase inhibitor. Also goes, the converse also is true. A lot of times we see that a certain pathway is not up, and then this standard treatment perhaps shouldn't even be done. And this is an example, this is just a visualization of where just one melanoma slide. So one field of view contains over 100,000 cells, and it has all these different regions, and they encompass from normal to precancer to the melanoma. And it contains different cell types that you can combine. So you have one tissue, but you can have, for instance, 7 regions and 5 cell classes. That would give you 27 proteomes, and then we can cut them. This is actually not such a big experiment either. This is how the AI sees it. So it's coloring all these different cell types, which we can then cut out. And then we have then we have these beautiful images. church window here. So this is additionally stained for this proliferation marker here. But we can just follow, for instance, the melanocytes over 5 regions until they become melanoma cells, right? And then it gives us a temporal evolution or quasi-temporal evolution of the cancer as it progresses to malignancy. But for the same person, and we also try to cut them out now for genome sequencing in parallel. So, but there's tons of information, so we can analyze all this by proteomics and then do bioinformatics and then back project that on the different regions of the tumor and do gene set enrichment and such like. So, for instance, we can look where's this famous or infamous Warburg effect happening, or any metabolic switch that these cells might be doing. We can see where that's the case or if that's the case. And here's a simple example here. So this is the skin again. This is the skin surface. This is the skin surface, But if you go further in, you have the same. So in both cases, you have melanoma cells, but they're different, right? And we all know that if they're further in, it's much more dangerous. So, and then we can say, what are these doing here? So for instance, in gene set enrichment, they are busy degrading the extracellular matrix, but these guys were not. They were instead more immune active here. So this could have different clinical consequences. So for instance, you would also choose to take 2 drugs, one targeting these, or maybe you only want to target these, or you want to even use 2 drugs to target the 2 cell populations. So now I'm switching away from cancer, and this is the example I'm most proud of, because we actually found the mechanism for a previously untreatable disease, but you may want to avert your eyes if you're not a doctor. So this is a melanoma, and this is Thierry Nordmann, who actually is a dermatologist, and this is drug rash. So every so often when you take any drug, even aspirin, you can have a very severe reaction, and this is even benign, and this is called DRESS, and this is called 10. And 10 is very severe, so fortunately it's also very rare, but it's like a severe burn, so people shed up to 1/3 of the skin, and also 30% of the people die. And it wasn't known, there was no effective treatment because people didn't know how does this actually happen. So then we have a relatively small cohort of healthy and these 3 different cases of drug rash. So we scan and segment and measure here, as I explained to you before, and then we can see so we can see this then in bulk. So in bulk, you don't get a good separation of these cases, and then we can resolve this into the cell types by DVP for the keratinocytes, so the skin cells, or for the immune cells. So this even separates best in the immune cells, which wasn't known at all. And then what we find is that we find an interferon signature. So this, by bioinformatics, this totally stood out. So all the top different categories between 10, this most severe class, and the normal, they're all connected to interferon signaling. And the good thing is that this is the JAK-STAT pathway, and JAK-STAT pathway inhibitors, for instance, against JAK1, they were already approved for the clinic in dermatology for atopic dermatitis. So then you can, and then we collaborated with one group in Japan and one group in Australia on mouse models of this disease, and indeed, so one in the WEHI in Melbourne and one in Japan, and indeed in both cases the symptoms resolved, and then there were 5 patients in China that have now been treated off-label with JAK inhibitor, and they all recovered. So within 2 weeks, they recovered, and you can see here that they've now formed normal skin here. So they are, as far as this disease is concerned, they're as good as new. Several of them actually, this arose as a complication of PD-L1 treatment. So now they still have the cancer, but they can be treated again with a PD-L1 inhibitor. So, so then, so this just shows, can we do this, or ask the question, can we even do this, if we had to, can we do this even in a single shape, if we really want to be like single cell, and indeed we can do that. So that was just published. So we characterized, so this is multiplexing now with DRA data, so we characterize the microenvironment with that in melanoma again, and then we, so this is where we want to go. So we do multiplex data independent analysis, and then we use this cell-in-one instrument for handling single cells and these single shapes that I showed you, and the setup, and we also put Astral, the new instrument that came out just recently to get better depth. And this is a paper that's also coming out now. So if we wanted to, we could actually do single shapes, and sometimes that's even necessary. So this is a classic case of zonation in the liver. So here we have the same cells, but they still have different functions. So, so what happens in the liver is you have the liver lobules, and then you have the hepatocytes organized circularly around them, or around the central, the portal vein, and they do different things depending on where you are. So we use this reference channel concept to even get close to 2,000 proteins, even from a single shape, which corresponds to a third of the cell. And then what we see is that the metabolic activity, this is also known, this changes with the relative distance from the portal vein. So there's metabolic specialization depending on how far you are away from the portal vein. So as I said, that was known. We also get all the markers that are known from deep sequencing, but we didn't appreciate that this actually switches so drastically for quite a number of these enzymes. And then this is, so we can then do machine learning, and we can ask, just from the picture, can we guess train what is the proteome of these cells, and we can do that to some degree. So that's the confusion matrix here, how good we can do this, and then we can turn it around and say, based on that model, can we predict the proteome just from the image information? And we can also do that to some degree. And this is my last example, but also cool, and from this is from organoids, so in collaboration with another group in Copenhagen. So they're working on organoids, and in organoids you can have the organoids in the human itself, and then you can, based on one cell, you can have an in vitro organoid, or you can transplant the organoid or make it grow in a mouse. Right? Now the question is, how similar are these cell types by function in these three situations? And proteomics should be able to answer that if we cut out these same cell types, and that's what Frederick did. So that's our workflow. So that's again the EvoSep, and with this new Astral. So the whole thing, this whole question with all the cell types and the three things is actually, so it was tricky to optimize everything, cut and so on, so the microdissection part, but the measurement was actually only half a week. So you can get all this data and answers in just a week, and we're getting very close or better than what you can get with transcriptomics. So we're getting closer to a depth of 10,000 proteins. And that's our goal, at least. And then this is what came out, so we can see how these cluster. And what we basically see is that these in vitro stem cells, they still have a lot of differentiation markers not fully expressed, and that means they need to be further matured. This is one take home. So with that, I'm closing, and so here we wrote a review how we think this fits into the bigger picture of spatial transcriptomics or spatial omics in general. And this is what I already mentioned. What would it take to make this single-cell proteomics biologically relevant? And with that, I want to thank my group and also collaborating companies and funding sources and so on.
Thank you very much, Matthias. This is an incredible journey of all the different applications of the proteomics. We have time for a couple of questions.
Thank you. It was an amazing talk. I was wondering, have we tried doing the correlation between the epigenetics and the transcriptomics of the single cell? And do we can see any relevant data in identifying the pre-cancer markers?
No, we haven't, so that's a good question. So maybe you meant to say epigenetics and proteomics?
Yeah, in a way.
Yes, so that would be very good. So we were actually, because we always cut out, so we thought, like, for instance, what I mentioned, getting the genomic changes, right? As we see the evolution at the proteomic level of the cancer, we want to see what are the mutations. And we thought, this would be easy, but it turns out there were no protocols for getting the genome of, from FFPE tissue. So we can do that now, but we cannot do this for, we cannot do a TAC-seq or something in these cells, but there are other approaches like Frank Wrong at Yale and so on. So we talked to him maybe. So we need to get that somehow together on maybe on alternative slices with this. So, but I think in general, this normally shouldn't get into an area where there's already some established PIs, but I think this is good for the next 20 years with all these combinations.
Yeah. Thank you.
Where do you see that you had a really great figure showing the increase in protein assignments over the last few years with this single-cell work? Where is there a clear sensitivity bottleneck somewhere that needs to be worked out, or is it just general improvements across the board that you can see this?
Yeah, there are tons of. You can see them as bottlenecks, or where we lose ions is also is an opportunity. So so I think. Digesting the single cell and not losing the peptides, that's relatively good now. So getting them into the mass spec. So where we spray them, this has been a challenge for decades. Do you really get all of them in? So as you lower the flow rate, you get more and more in, but probably you lose an order of magnitude there even. And then the way with data-independent acquisition, particularly on this new Astra, we still lose a lot of the ions, right? So they like to use small windows. Maybe they cover 1% of the ions at any time. So again, there's an upside of 100-fold, or that's why we are stuck right now. Yeah, but the median protein in HeLa cell is 18,000 copies, right? So one should be able in the far future to go to 10,000 or complete proteome even in a single cell.
No? Okay, let's thank our speaker once again.